Hello, everyone. My name is Foon. I'm the wet lab instructor, and I'm going to give you a little break from Holly. I know it's been a while since you've seen me last, so welcome. I'm excited to be sharing with you a little bit of my world in the next week or so with you and give you a little break from computational stuff, even though that stuff is pretty cool. I have done so wet lab means that it's experimental work that's done on the bench. So I'm working with chemicals, tissues, samples. I'm in the bench doing the work, very hands-on stuff. I love wet lab. I've been doing wet lab for, I guess, over a decade now, which is crazy. I started my freshman year. So the summer after freshman year, I joined an REU program, which I highly recommend anyone who wants to get into wet lab. You should definitely check out REU programs at different universities. I think it stands for Research Experience for Undergrad. So they're summer programs usually that's specifically geared towards undergrads and specifically to introduce undergrads to wet labs. You kind of like get matched to a lab and you do work for the lab for the summer and you get trained. And that was how I got my first official lab training. I mean, I did some lab work in high school, like, lab classes and all that, but I never actually worked in a a research lab until undergrad. So yeah, I started the summer after my freshman year and really have stayed ever since. I've worked with a lot of different models. So I've worked with in vitro work, which means like cell work. I've also worked with ex vivo, so that's more like tissue work, right? And that's actually what we do in the NANS lab. We work with brain tissue that's kind of taken outside of the body and grown on in like dishes and grown in like on membranes outside of the body. So that's what ex vivo means. And then I'm slowly being introduced to in vivo work, which means like within the body. So I've been working with a collaborator on working with vaginal samples. So our samples are from human human tissue. And I've also been working in terms of in vivo work. We've been applying our special treatments to mice um, in vivo. So we've been doing surgical procedures on them and stuff like that. So I've worked with all across the spectrum, honestly, from in vitro to ex vivo to in vivo. And I really love experimental work. And I hope to share a little bit of that excitement with you in these upcoming modules. These modules are what I think are really important skill sets that we learned specifically in the NANS lab. But to be honest with you, I've used these skills at other labs too. So wherever you end up working, if you're interested in experimental work, these skills I believe are important to you. So we're going to today talk about immunofluorescence and staining. And then the next module, we'll talk about confocal imaging and just imaging in general, honestly, of samples. And then lastly, we're going to work with different experimental models. And that really brings together, I think, the mindset that you would take when you go into an experimental lab for the first time. So yeah, if you have any questions, feel free to Slack me, of course. And I will share my screen momentarily and we'll get started on this presentation together. But yeah, and I'll probably take my camera off just because my Wi-Fi is a little spotty and it doesn't work super well if I have both my camera and the screen share on. So I'll turn my camera off. But again, if you have any questions, let me know in the Slack and I'm happy to answer them. And I just want to point out, even if you're not interested in experimental work, or even if you don't think you want to work in experimental-based lab, I believe it's still really important for computational like scientists and researchers to understand where their data comes from. So, you don't, as the data that you analyze came from somewhere, and it's probably an experimentalist who's collecting that data for you. So understanding where the data comes from gives you a better appreciation of the work that's put into collecting that data for you, and also just what variables and what unknowns are present in the data as well. So just understanding the experimental process can really help your workflow, even if you're a computational person. So yeah, I hope you learned something today. And I'm just gonna turn off my video and then share my screen. All right, so let's just get started on Module 1, the basics of immunohistochemistry and cellular staining. I'm going to be using a couple of really pretty images throughout this whole presentation, and I think that's the exciting part of immunohistochemistry and cell staining, that you get to be able to actually image these cells that are microscopic, which is really amazing, I think, to me. And that's really what drew me initially as a freshman to experimental work, because in books we've seen pictures of cells and everything, but I actually got to see what the cells looked like in my samples in real time. And that was a really great experience for me to really understand how the cells were organized and what they actually look like under a microscope. So yeah. All right. So this is something that you'll be doing in the little like Google form, but I just want to get you all started thinking about how these cells are visualized and how do we really know what cells look like? If you think about it, it's really amazing that we can actually have the capability to image cells now. So, these organisms are microscopic. You would not be able to see them with the naked eye. And there was a time when we had no idea that what the cells even looked like, or that tissues were made up of cells at all. So cells really make up your entire body, and it's really amazing to be able to see them. And it's really cool also because different cells look differently depending on their functionality. So for the pre-module activity, I would like you all to look over pictures of central nervous cells from any paper and note the different structures and colors that are presented. So you can see here, it's just two images um, taken in the lab by me and my undergrad, Jimmy Yi. And you can see that there's two different cell types and different colors represented here. So there's the green on the left, and that's microglia. You can see there's a little bit of branching, which microglia are known to do. And on the right, there's a couple colors. You have the blue, which is DAPI, and that stands for the cell nuclei. And then there's also the red, which stands for astrocyte. And you can see the red has very extensive branching in this photo, and that's really cool. And that's representative of the different functionalities that microglia versus astrocytes have. Yeah, and I just want you to, throughout this activity, just really focus on what colors, what cell types do you see in the images, what type of structures do you see stained? Do you see branches like the branches that I showed you previously, or do they stain for like organelles within the cells themselves? So keep in mind the structures that were stained, and then specifically what dyes and stains that these researchers use. So yeah, have fun just looking around at papers and looking at really pretty pictures, basically. Okay, so the heart of immunohistochemistry is the immuno part, which refers to your immune system, specifically antibodies. So antibodies are present in honestly everywhere in your body. There's a lot of antibodies, and these are just a couple places you might not actually think about antibodies being, but there's a lot of antibodies in your eyes, because of course your eyes are open a lot of the time, and that makes it really easy for uh, like other microorganisms to get into your eyes. And so there's a lot of high concentration of antibodies in your eyeballs and in your tears. And antibodies are basically your body's immune defense, like your body's defense system against any invading organisms that might get you sick. So there are a lot of antibodies like in your mouth as well, because you eat a lot of stuff. You're ingesting a lot of things from the outside world. And so, your body has to protect against those microorganisms. Similarly, if you get hurt, like scrape your knee or something, antibodies uh, are really principal components of the response to healing and your body's response to attacking any foreign invaders that might have entered through the wound, and also healing those wounds as well. So this is what an antibody looks like. It's like a Y shape, and there's two different regions. There's the variable region, which changes depending on which microorganism the antibody targets, and the constant region, which, as you can, it's kind of self-explanatory, but it remains constant between different antibodies. Now, and this variable region is the part of the antibody that's responsible for attaching to foreign microorganisms that might want to invade your body. So here you kind of see that there's like a little shape, on the antibody itself. Let me try to turn on my laser pointer. Okay, here we go. Yeah, here you can see on the very tip, the shape, there's like a little indent. Specifically, it's like a semicircle indent. And this circle fits perfectly onto these uh, epitopes, is what we call them. So they're like basically signatures of the microorganism. So these epitopes on these foreign invaders that your body might be trying to defend against. And antibodies are also specific. So they'll bind, for example, this one in particular will bind to around their epitope versus this epitope, which is more triangular. The antibody would not bind to that, for example. And yeah, they're also specific to what type of what type of epitope they were created to protect you against. So there's two different types of immunofluorescence staining. There's direct and an indirect. So direct means that, as you can see, there's that Y antibody right here. And this green, I guess, hexagon, I had to guess, I forgot the words for that. This green hexagon is a fluorescent molecule. So this is the fluorescence is actually how your eyes are, how your eyes see what the antibody is attached to. So for direct staining, you have your antibody bound to the fluorescent molecule, and that binds to the red triangle here represents like an epitope, for instance. So that's direct immunofluorescence staining. The antibody binds directly to the epitope, and the antibody also is fluorescently marked. So you can visualize the sample that way. Now, indirect staining requires two different antibodies, as you can see, and they're kind of stacked on top of each other. You have this blue one, which you call the primary antibody, and that's the one that binds to the epitope. And then you have something called a secondary antibody, which binds to the blue one. And the secondary one has the fluorescent marker on it, so you can image the sample. Now, as you can see, there's differences between each one. The direct one is quicker because you don't need two, you only need one. But it's also more expensive because what that means is that you need to create a different antibody for every, or you need to, the fluorescence molecule is is expensive, basically. And so that means you have to have a different fluorescent marker for every epitope that exists, which gets really expensive. Whereas for indirect, it's usually cheaper because, as you can see, the fluorescence marker is bound to the secondary antibody, which is then bound to the constant region. Do you see how it's bound to the constant region of the Y here? It's bound to the constant region of the primary. And the constant region is constant. So a lot of different antibodies have the same constant region. So you can just create one secondary antibody can bind to several different types of primary. So you don't have, there's not that much variety as there is for direct. And because of that, it's cheaper because you can use the same secondary for a lot of different primaries. There's also higher sensitivity, amplification, and multi-target detection using the indirect. So that's the primary. We use both types in the NANS lab, but we use indirect antibody staining more frequently. And then, yeah, this is what I meant by amplification and multi-target detection. You can have multiple secondaries bound to the same primary to kind of increase that fluorescence so you can see this sample brighter. Now, you might ask, how do you know which antibody to use? And honestly, that depends on what you're trying to find. There's a lot of different applications of immunofluorescence staining. So if you have like a specific protein you're interested in, you can find through immunofluorescence staining, you can figure out the expression level of the protein, whether it's intracellular or extracellular, and where that protein is going. You can kind of track that protein as well over time. And so that's really cool. You could also, like, if you're interested in a larger scale, so not proteins, but if you're interested in cells themselves, you can certainly stain the cells as well. And you can figure out how many cells are, the cell density, how thick something is. Like if you have like a tissue and you want to know how many cell layers something has, you can stain to determine that. You could also think about what else. You could also think about like what you're culturing the cells in. So whether or not you're culturing them on a plate or in a gel or I don't know, like on a more 3D model, you can figure out what type of staining will work best for each situation, as well as the number of samples that you have. So all of these things, all these design parameters uh, help you determine what type of antibody you need and what, I guess, whether or not you want to use a direct or indirect method. So the first step of staining is cell permeabilization. It's a hard word for me to say, but basically this permeabilization allows antibodies to enter through the membrane and access intracellular epitopes. So again, you have cells are protected by a membrane, and you need to, whatever you want to stain, usually it's intercellularly, so it's within the cell. So you need to actually be able to get through the cell somehow. So you use these reagents to help break down the outer cell layers so you can actually get your antibodies in the cell itself and stain what it needs to stain. So there's different reagents out there that you can use. In the lab we use Trypan X100 or Queen 20, which is less harsh than PBS. These are just detergents, so they break down the outer layer of the cell. You can also fix cells using alcohol. So methanol fixed samples are already permeabilized, and alcohols can easily wash out the lipids of the cell membrane, however. So if you're interested in, like, here's an example of a cell membrane with a lipid. If you're interested in these lipids, you might not want to use a methanol or any alcohol fixed samples because it can really wash out these lipids. And so it's probably better to use another reagent instead. And again, a lot of wet lab is just optimization because you're working with different samples, you're working with different tissues and conditions, and you have to find out which antibody works the best, right? So this will all depend on your cell type, what cell type you're using, what you're looking for, like what epitope you're looking for, and what antibodies you have present in the lab. And depending on your sample, you'll also determine what type of permeabilization agent you're going to use. So again, if you're interested in lipid samples, you might not want to do alcohol, for instance. So that all depends on your sample design. So now, after you actually permeabilize the cell, you're going to have to block it. So blocking helps minimize the intra or extracellular background signals, which reduces nonspecific antigen antibody staining. So I think a lot of antibodies are they can be not very specific. So even though they're supposed to bind to a specific epitope, sometimes they just by chance bind to other things that aren't what you're looking for. And so in order to reduce that non-specificity, you actually need to block your cells in a blocking reagent, which is often the serum of the host in which the secondary antibody is made. So for example, if you're using a goat anti-rabbit secondary antibody, that means that your blocking buffer should contain goat serum. And that's because, and I know this is a little complicated. I think this is the hardest part of cell work or in immunofluorescence staining, because there's two different antibodies. You have, or there's two different types of animals that you're using, right? You're using goat and rabbit as well. So we're going to go back to the slide with the indirect stain, so I can just go over this with you. So yeah, right now we're only going to focus on the indirect staining, right? So the primary antibody binds to your epitope, right? And I just want to keep in mind that antibodies are derived from animals, and we have antibodies within our bodies as well. So different antibodies are called different names depending on what animal they are derived from. And so for, let's say, for example, this blue one is from a rabbit. We call it anti-rabbit because it's an antibody that's from a rabbit. So we call the blue one anti-rabbit. And then say the yellow one, the secondary, is from a goat, for example, because the goat, we need it to be a different species, because if it's the same species, the antibodies from the same species don't attach to each other. You need the secondary to be a different species than your primary for it to react, for it to bind to each other. So for our lab, we like to use goat to bind to the rabbit, for example. So in this example where the blue, the primary, is a rabbit, and the yellow, the secondary, is the goat, for the blocking buffer, we want to use a goat serum, because then if we use a goat serum, again, antibodies of the same species don't bind to each other. So if we use a goat serum, then the secondary antibody won't bind to the serum itself. And you don't, therefore you reduce any background and non-specific binding. In that situation, if you use a goat serum with a goat secondary antibody, that the secondary antibody will only bind to the primary, and it won't bind to any like external, like anything else that isn't the rabbit, basically. So that's kind of an overview of what this primary and the secondary antibodies are. And I know it's confusing, but if you just go back, and all you really have to keep in mind is just understand which animal your primary came from. So in this example, the primary came from the rabbit. That's usually the first kind of this inner species here, anti-rabbit. That means that the primary antibody is from a rabbit, and the secondary antibody is a species that's different from the primary. So just keep that in mind. And I know it's complicated, but as long as you know what species your primary is and your secondary is, then you're golden. And I'll go over this in the next lesson, but AF 488 stands for electropluorophore. As you can see in the name, it's a fluorophore. So that's for the imaging, that's fluorescence. That's the fluorescence molecule right here. These green hexagons, these represent fluorophores, and that's what's going to help you image. But we're not going to talk about that in today's lesson. So you don't have to worry too much about that. Just know that this is the fluorescent. The secondary antibody is attached to like a fluorescence molecule. So this is just going into depth about the process. If you join a lab and you have to do staining, this is the type of process that you're going to go through when you want to start incubating your samples with the primary. So you want to make sure that you choose a epitope that has high specificity, and you want to be able to choose a antibody that binds that epitope as well with high specificity. And there's a lot of different antibodies out there. Again, there's a lot of different animals, but there's also a lot of different types. So there's mono, polyclonal, or recombinant. And that basically, here's the different examples here. It basically tells you like how many different things the antibody can bind to, for example. So that all depends on what you're looking for. So for example, in the NANS lab, we have a lot of studies on microglia. And microglia have this unique molecule called IBA1. And so we use, so in this picture here, to pretend that the yellow, the epitope is IBA1, right? There's several different antibodies that bind to IBA1 actually. So we have to choose which one works best for our experiment. So that's something that you can just think about when you're designing your experiment. And then these are things that you'll probably work in experience if you ever work in a lab that does staining. A lot of the optimization is about antibody concentration and incubation time. So how much antibody do you use and how long do you kind of leave the antibody to interact with the cell? These are all questions that you're going to work through and optimize in a lab. So after you have your primary antibody incubated and the primary antibody has gone ahead and bound your epitope you're interested in, then you're going to conjugate that with a secondary antibody. And as I said before, a secondary antibody has to be in a different species than the primary. And what you do is pretty straightforward, similar to how you incubated the primary antibody. You just add the secondary antibody in your solution, and it kind of just goes where it needs to go. Honestly. So you incubate your whole sample with the secondary antibody. And the secondary antibody does have that fluorophore. So this is how you're going to go ahead and image your samples later on. And then usually the incubation is 2 hours. But again, I'm going to go back to the previous slide. Again, incubation time is something that you optimize. So depending on your sample, the incubation time can change. But in our lab, it's usually 2 hours for the antibody. And then after that, you wash with PBS, which is a buffer. It's basically the salt solution, basically, and you wash your cells. You wash you wash off any secondary antibody that didn't attach, that didn't bind to a primary. Any excess secondary gets washed away, and that reduces any background signal. Now, I will talk about this in tomorrow's class, or not tomorrow, but like later in the class next week, the next class. But a fluorophore is how you're actually going to image the samples that you stain. And a fluorophore is basically a chemical molecule that's used to label different structures for analysis. So fluorophores are excited when they absorb light of a certain wavelength, so they get excited here. The molecule raises energy and it goes up to the next energy level. And then once it loses that energy, it goes back down, it releases some light, basically. It releases the energy in the form of light. And that's how we are able to see and visualize these cells. Yeah, so don't worry too much about this plot. I will go over this in the next lesson, but just know that these fluorophores that are attached to the antibodies are really important for us to actually visualize anything. And it's kind of this combination of the excitation and the emission once the fluorophore falls back down to the ground state that will allow us to see the energy in the form of light. Now, this is what is also important for immunohistochemistry, counterstain, and mounting. So you have cells, right? And the counterstain helps you determine whether or not what you're looking at is actually a cell. So in this example here, if you remember earlier, the yellow, we're interested in microglia, specifically IBA1 epitope. So the IBA1 is unique epitope for microglia. So the primary antibody binds to the IBA1 and the secondary antibody binds to the primary. But how do we know that it's actually in a cell and the IBA isn't like outside of the cell, or like it wasn't some sort of contaminant outside of the cell? Well, that's what the counterstain is for. So a popular counterstain is DAPI, or host, and it stands for the nucleus. So when you see a sample that has both the DAPI, so the blue, and the IBA, which is usually green, when you see a sample that has both of those at the same time, that means that it's most likely a cell. But if you see some IBA staining, so if you see something that's like green but doesn't have a nucleus as well, that probably means that it's like some sort of contaminant or it's non-specific binding. So a chemistry is really important to determine like the morphology of the cells or its cell structure and just target localization. So make sure that what you're staining is actually like within the cell. And lastly, you're gonna mount your cells. So you don't want your cells to slide around or anything when you're imaging it. So you want to make sure that you put the cell in a, usually a buffer that has low autofluorescence, so it doesn't interfere with your imaging. It prevents your sample from drying out, and it also stabilizes the refractive index. Again, this all just improves the imaging. So you know that what you're imaging is actually the antibodies and not something else. So we're going to go over this in the next lesson as well. But for the NANS lab, we do work with confocal images. So we use a big microscope called a confocal. And this confocal is able to detect a lot of different wavelengths of light, which is really great. And because of that, we can often capture these images of different cells and different target epitopes that we're interested in. So here are some examples from our lab. Again, you can see that DAPI is a popular counterstain. So we want to make sure that the samples that we are interested in have both DAPI and IBA1, which stands for microglia. So we can know that these are actually cells and they're not like some random contaminant. So yeah, you can see there's 3 different types of stains that we use. The counter stain is the same for all of them. So these 2 are DAPI. I guess the host is also a nuclear stain. So they stay similar to DAPI. It's blue. It stains for the nucleus. But here in this image, we're staining for IBA1, so microglia. This one we're staining for OLIG2, which is OLIG dendrocytes. And then the lyso tracker stains for lysosomes. So this is an organelle-based stain, similar to host, which is the stains the nucleus, which is also an organelle. So lastly, I just want to close out by saying that there's a lot of different applications for staining. And I know that a lot of my examples so far have been specifically for cells, but you can absolutely do it for larger things like tissues or organs, for example. So immunofluorescence is commonly used to determine the expression of different antigens, epitopes, both intracellularly and on the membrane itself of the cell. It can also be used to identify the presence of like different bacteria or viruses or other parasites within your sample. So if you want to know if your sample of cells is contaminated with a bacteria, you can go ahead and stain for that bacteria and look under a microscope and see if both the cell and the bacteria show up at the same time. Because if they do, that means that you have bacteria in your cell samples and you should probably throw those out. And this is just examples of pictures of different viruses that are within a sample. In the NANS lab, we also use antibody staining to track the cellular movement or cell behavior. So again, once the secondary antibody is attached to the primary, it sticks there. It doesn't move. So you can watch different spots of fluorescence kind of move as the cell moves, and you can capture movement that way. We can also label non-cellular structures such as polymers or gels, for instance. So if you have specific proteins, you might recognize this as an electrophoresis gel. You might have done this in high school, but each of these bands here, the dark bands, represents a different protein. And these proteins, you can use antibodies to stain for these proteins. If there's like a specific protein that you're interested in, go ahead and use an antibody on it and stain for the one that you're interested in. And then you can be able to see whether or not that protein exists, like whether or not you have that protein in the gel. And then I like to close out by using common application of, I guess, antibodies, which is pregnancy tests. Of course, there's also COVID tests, but I'm sure we've all heard about those. And so I like to talk about something a little different, which is pregnancy tests, which uses the same type of idea. So you have your sample here. that usually urine sample on a sample pad. The urine sample moves throughout the device. So as it moves, it picks up these antibodies, which bind to HCG. So HCG is an epitope that identifies whether or not you're pregnant. So the sample binds to these free moving antibodies, these primary antibodies, HCG, and it moves through the first test line. And the first test line has a secondary antibody that also binds to HCG. So what you'll get is this sandwich structure, and there's a reaction that occurs. In this case, it's not a fluorophore, it's not fluorescent because you don't need a laser to see it. It's not a fluorophore, but when this reaction happens, you'll be able to see this dark line that changes. So it's like a different enzyme that changes. It's an enzyme on the antibody that changes color when there's something's bound to it. So as soon as something is bound, as soon as the HCG marker is, which is what you're looking for, binds to this antibody, it turns, and you have the sandwich structure, it turns a darker color. And that's how you get these lines, for example. And your sample just goes through the device this way. And I guess what's important to take away from this is that antibodies can be used to not only image cells and tissues, but you can also use them for these tests if you want to identify whether or not an epitope is present. So if you want to identify whether or not you have a target protein, target protein, you can go ahead and use antibodies to be able to label these specific proteins that you're interested in, and then be able to have some sort of readout, whether that be fluorescence or whether that be like an enzymatic change that causes a change in the color of these lines, for example. So the COVID test works similarly. And I just want to close out by showing these really nice pictures of different tissue structures, different cell structures that are generated with immunofluorescent images. So yeah, thank you so much for joining today. I hope you learned something about immunofluorescence and immunohistochemistry. This leads really well into my next lesson, which is about confocal microscopy. So how we actually image and see these cells and these structures that we're labeling for. So I hope you're excited for that. Otherwise, have a great rest of your day, and I'm looking forward to hearing your responses in the Google Form module. Bye.